Hello, hi. Thank you so much, the organizers, for inviting me to give a talk here at the Keystone. And my talk will not be focused on the single cell techniques, but rather how we have used single cell techniques to learn more about a system that it's not an organ, but it's a system that connects multiple organs, and that's the sympathetic nervous system, of which we see an example here. So essentially these neurons here that are labeled in red. And I'm also going to talk about neuroimmune interactions with these nerves that in the end, they will control the fate of the adipose tissue. So the tissue where we store fat and which determines when it's big, if one is obese or not. So body weight homeostasis, it's largely controlled by a hormone leptin, which is produced by the adipose tissue in amounts that are proportional to the amount of fat. And in this way, the brain knows how much fat exists in the body. So the more fat exists, the more leptin exists in circulation. And this is the afferent arm in the neuroendocrine loop in which the brain senses leptin and therefore knows how much fat exists, which is produced by the adipose tissue, which is in fact a gland, an endocrine gland. And once there's enough fat, the brain triggers a plethora of responses that includes suppression of food intake, which in turn reduces the amount of adipose tissue. Once the adipose tissue is smaller, then it produces less leptin, and somehow less leptin leads to increased food intake, which then makes the clock to tick the other way around. And for a long time, it was thought that the only efferent mechanism of action of leptin was to decrease food intake. However, there were experiments suggesting the opposite, that initially, and these experiments are called pair feeding experiments. Pair feeding experiments are experiments in which we have an animal that is obese. It's obese because it's deficient of the hormone leptin. So it doesn't have leptin because of a monogenetic mutation. So the brain senses that there's no fat, and then it just triggers mechanisms to increase fat mass increase, so obesity. And these animals are hyperphagic, they eat a lot. And when we cure these animals, and also humans with the same mutation, with leptin, we see that there's a linear decrease in body weight. So these animals, they lose all the fat they have. And if this effect was solely mediated by suppression of food intake, then one would expect if we would be feeding another subset of animals that was not treated with animals with the same amount of food that leptin-treated animals would eat, then one would expect similar weight loss. But the fact of the matter is that only happens in the beginning, in the first few days. And after that, the animals that are pair-fed to leptin-treated animals but do not receive leptin treatment, they don't lose the same amount of weight. So there is a delta here. And this is not observed just once. It was observed by multiple groups in different institutes. So we thought, what is that is generating this delta? So this was the question that led me, or this was the question that inspired the beginning of my lab back in 2013 at the Gulbenkian Institute in Portugal. One glimpse into this, into the potential answer to this question came from the observation that leptin deficient patients, they have what is called orthostatic hypotension. So this is when a patient lifts up from the chair, it's going to feel a transient decrease in blood pressure. And this is thought to be regulated by sympathetic nerves that innervate the vasculature. And there's also another deficient response that these patients have, and it's called the cold pressure response test. When people put a hand in a bucket full of ice, one's going to see a deflection, a transient change in blood pressure, which is measured here in the arm cuff on the contralateral arm. But leptin deficient patients, they have abnormal responses, although they have normal blood pressures. So this is a deficit in response to challenge, whether gravitational or temperature. And this is thought to be mediated by nerves that innervate the vasculature. And I thought that perhaps sympathetic nerves that would innervate the adipose tissue would be responsible for that delta. But the fact of the matter is whether or not sympathetic nerves innervate the adipose tissue was a topic of contention. There was just no clear answer because there was no primary data that would be sufficiently convincing. And most of the textbooks, still to these days, they simply neglect the adipose tissue, or at least the older books that have been published prior to the last 5 years. So to address the question of whether or not the white adipose tissue is innervated, because it was known that the brown adipose tissue was well innervated, and there's primary data that is very convincing. But whether or not the adipose tissue was innervated, this was a question that we addressed not by classical histology methods that yielded data that was not very convincing, but rather by using techniques such as the clearing techniques. And in these techniques, we have a piece of fat, which is a whole adipose tissue from a mouse. In this case, this is the inguinal adipose tissue. And through a series of chemical steps that were developed by the lab of Marc Tercier-Lavigne at Rockefeller University, we were able to render this piece of tissue transparent. And if we couple this to an antibody staining that marks sympathetic neurons, and then we're able to see that the adipose tissue in a whole mount preparation that was rendered transparent, we can clearly see that there's a vast network of axons, sympathetic axons, that are labeled here against tyrosine hydroxylase, which is one of the enzymes in the biosynthetic pathway for catecholamines. And as you can see here, this is pretty convincing that these are sympathetic axons. Now, you can imagine that if you were to do an optical slice on this image, you would just have dots. And of course, those dots are not very convincing. And that's what classical histology techniques would enable you to see. Now, one could say that these nerves that are innervating a slab of adipose tissue, they're there just to subserve the biology of the vasculature. So we decided to follow these nerves, this time around with a different imaging technique. This is to photomicroscopy in an animal that expresses a fluorescent reporter in downstream of TH3. And we see here that the adipocytes that are labeled here in green, they're being subject to targeting by these sympathetic neurons. So now we can convincingly or be a bit more convinced that in fact the adipocytes, they do receive neuronal input from sympathetic nerves. Now, textbooks, they knew for a long time that adipocytes, they have the receptor that sees norepinephrine, and that norepinephrine is a neurotransmitter that sympathetic neurons release, and that we know that downstream of activation of the adrenergic receptors in adipocytes, we have lipolysis, which is the breaking down of fat. What was not known is whether the source of norepinephrine that underlies homeostatic changes in body weight and in fat mass are these neurons and not something else, for instance, the adrenal gland. The adrenal gland also produces enormous amounts of norepinephrine that enter circulation, but that only happens in a fight or flight response. So that is when you have to run away from a dangerous stimulus, or if you do surfing or parachuting or so-called adrenaline sports. It doesn't really happen if you skip a meal, or if you skip several meals, in fact. And moreover, animals that do not have adrenal glands, or humans that are injected with leptin, they can still undergo homeostatic changes in weight. So if it's not the adrenal gland, what is it? Is it these neurons? And this question bodes for a gain-of-function experiment in which we utilized optogenetics. We expressed chanoordopsin, which is a light-activated cation channel in these neurons. And we, through the implantation of an optical fiber subdermally, pointing at these neurons, we're able to activate these neurons and demonstrate that optogenetic activation locally of sympathetic neurons in fat is sufficient to drive fat mass. And through a series of experiments in which we also utilize genetic loss of function experiments as well as surgical, we demonstrated that the sympathetic neuroadipose junction mediates leptin-driven lipolysis, and that in turn they close the neuroendocrine loop of leptin action in the brain. Now, at homeostasis, the clock ticks perfectly. There's an afferent arm, there's an efferent arm, and it ticks such that it keeps weight in a very narrow range of variation. But during obesity, there's a phenomenon called leptin resistance. And in leptin resistance, the hormone, there's increased levels of the afferent arm, that is to say, of leptin, so there's hyperleptinemia. But for some reason, these high levels of leptin, they're not sensed by the brain. And therefore, there's an efferent response that it's reduced. So the idea of trying to create a mechanism or a therapy that would strengthen or amplify the efferent arm was a question that I entertained in my lab, because there was already evidence that especially from the lab of Philip Scherer at UT Southwestern, that in order to revert this equilibrium back to homeostasis, it is sufficient to tweak with one of the arms in the neuroendocrine loop. He demonstrated that by reducing leptin levels, then one can rescue normality or can mitigate obesity simply by interfering with one of these arms in the neuroendocrine loop. So Philip Scherer did the afferent arm, and I decided to do the efferent arm. And this idea would be simply that if one would be able to increase the tonic activity of these neurons or facilitate the activity of the neurons that it's lost in a leptin-resistant state, then we would be able to zap the fats in the white adipose tissue. And we could think of two alternatives. One could design a drug that directly targets these neurons solely in the adipose tissue, which is the more difficult part. Or one could perhaps target a drug to cells that would be talking to these neurons. And this is when we started studying these cells, because we believe that perhaps if these beautiful cells here that we now know are sympathetic associated macrophages, if these cells are in direct contact and if they negatively regulate the sympathetic neurons, then we would be in a good position to target these cells with immunotherapies. And to make the long story short, so that I finish up my introduction and I jump onto the single cell data. This is the role of the cells that I've just showed you. These are sympathetic associated macrophages, and they have specialized machinery to deal with norepinephrine, which is the signal that the adipocytes need to see in order to shrink and become smaller. In a situation of obesity, there's a lot more of these cells that get recruited onto nerves. And we demonstrated that abrogation of the machinery for norepinephrine uptake by these cells, abrogation of this machinery utilizing the tools of immunology, which rely on bone marrow transfers of knockouts for these transporters. So you can think about these cells as little vacuum cleaners, or in UK we call these hoovers. That have a transporter, and the transporter is the solid carrier 6A2. And by doing a bone marrow transfer of a knockout, a whole body knockout, by transferring the bone marrow, which we demonstrate to be an approach that it's sufficiently tissue specific, because no other cell in the bone marrow lineage expresses the norepinephrine transporter other than the cells that we identified. We demonstrated that this approach that spared the brain, because the bone marrow transfer cells do not repopulate the brain, but they do repopulate the immune system and lineages that derive from the bone marrow, which is the case of the cells. We demonstrated that this brain-sparing approach that is sufficient to mitigate obesity. So this gave us the idea that perhaps if we would have a drug that would function like this cork here in this cartoon, we would be in a good shape to help patients. And through a sinuous path, we ended up developing, modifying a classical norepinephrine transporter blocker, which are amphetamines. And this is the graphical abstract that, to make the long story short, we modified amphetamine by pegylation, and we demonstrated that this drug does not enter the brain to phenocopy the phenotype that we had seen in our previous approach. We demonstrated that not via the transporter, in fact, but via the beta-2 adrenergic receptor, a pegylated amphetamine drives weight loss because it promotes thermogenesis coupled to heat dissipation. And because it spares the brain, it also spares any cardiotoxicity, which is usually associated to intake of amphetamines. So this thermogenic activity is also one of the one of the energy dissipation mechanisms that is known to be part of the efferent axis in the neuroendocrine loop of leptin axons. So we demonstrated that sympathetic neurons, they drive lipolysis, which makes the leptin-producing gland, that is to say the white adipose tissue, smaller. But downstream of this lipolysis, we don't have free fatty acids circulating around. We actually, the hormone in its natural state, or on the homeostatic state, it actually drives activation of the brown adipose tissue that in turn burns all the fuel that it gets released from the white adipose tissue. So white adipose Okay, so the sympathetic neurons that are innervated, in fact, by this ganglia, or the brown adipose tissue is innervated by the superior cervical ganglia, which also have SAMs, sympathetic associated macrophages. So we asked, is it the case that there's other immune cells populating this sympathetic ganglia? And through an heroic effort by Vitaly Sarkar, who is a Novo Nordisk postdoc in my lab in Oxford, she was able to dissociate single ganglia and dissociate enough of them so that we were able to run, in collaboration with Novo Nordisk, single cell sequencing of these neural populations. And as you can see here, we see that there's a plethora of immune cells that live in this ganglia. And in fact, the neurons, they are actually not the majority, but I think this is a technical artifact because these neurons are very big and they're like balloons full of water. So they're very sensitive to damage, as is known in neuroscience that neurons are complicated and difficult to isolate. But see here that there's a frank population of immune cells, and we have faithful validation that we don't have cross-contamination. So we have faithful markers. We see that immune cells are only marking immune cells. They're not marking neurons. They're not marking parasites. We see that there's here faithful marking, and it's a really beautiful data set. I think that reveals high technical quality in the hands of Vitaly. And we asked whether or not if this data set could give us any links or any views on what would leptin be doing to immunometabolism. As you may know, immunometabolism is a growing field, and the Keystone has had several meetings in immunometabolism. But immunometabolism studies mostly immune cells in the adipose tissue, and very rarely is mentioned that immune cells are actually creating any link with a well-known controller of adipose tissue, which is leptin. So we looked into this data set and we curiously saw that the leptin receptors seem to be expressed in this subpopulation of cells, the endothelial cells. It seems to be faithfully marking this population of cells together with other markers of endothelial cells. Of course, it doesn't mark the neurons, and the neurons are marked by faithful markers such as TH. And then when we look at the histology, we saw that these sympathetic endothelial cells, they are in fact perineural barrier cells. So we can see here, it looks like a sleeve. If when you dissect sympathetic axons or nerve bundles that contain axons and also other nerves, we see that there's this sleeve-like structure. And we can look at it in different ways. So if you do a cross-section, this is how we look. They really look perineural. This is not a blood vessel. They are endothelial cells, but this is actually perineural cells that are very thin and are populating ganglia and sympathetic nerve bundles. We also did immuno-EM, and we saw that this very thin layer of cells is actually very thin, and they seem to be perineural. So we asked whether or not these endothelial cells, they would be acting like other barrier cells that exist in the gut or any other structure. So we saw that IL-33, which is well known to be a cytokine that it's produced in barrier areas in the body, it's also produced by these perineural cells. And we know that IL-33 is attractive for Tregs, and we see that the Tregs, they seem to be devoided in the brown adipose tissue of animals, conditional knockouts in the leptin receptor in these perineural cells. We see that there's a deficiency in thermogenesis, and we also see that these animals are prone to a high-fat diet. So altogether, it seems that the IL-33 in specs, they confer protection for obesity independently of food intake. And this refers to the literature that already have identified neuroimmune interactions with the Tregs and macrophages in the brown adipose tissue. And we think that finally the missing piece in immunometabolism puzzle that links leptin to immunometabolism are the specs that seem to be connecting these two different fields. So the leptin field and the immunometabolism field. The initial work was done at the Gulbenkian Institute, mostly by Inez and Roxana and Elsa. And I already mentioned the people in Oxford who took part of the spec work. And thank you so much.
Thank you very much, Anna, for this interesting talk. I have a question about the signals that cause the recruitment of the macrophages in obesity. Are these signals coming in from the adipocytes or from the neurons, or why do we have more macrophages in the obesity?
Well, we don't know yet. So this is a question that we're actively working we still don't know what's the macrophage recruitment signal. So it's an open question. We're working on it.
Great, thank you. And then you have sort of a three-way, like a triangle of the neurons, the macrophages, and then the adipocytes, and the endothelial cells coming in the middle. So did you do any kind of formal sort of ligand receptor analysis on your single-cell RNA sequencing to try to figure out who talks to who here in this?
Not yet. This is actually a very good suggestion. We haven't done that yet, but of course, I think Italy would be interested in these sorts of analysis.
Cool.
Thank you.
Thank you very much, Anna.
Thank you.